Welcome back to the Deep Dive, where we take your source material and extract the most important nuggets of knowledge. And today we are diving deep into a fascinating question about, well, nature's smallest, most high-octane flying machine, the hummingbird. You know the drill, their primary fuel is this super sugary nectar. But here's the surprise factor, the thing that shifts this from Biology 101 to something really unexpected. Nectar, whether it's in a flower or a feeder, is often naturally fermented. So this brings us to the question that I think all of us are thinking. Do hummingbirds ever, catch a buzz? Do they get a little tipsy?
That is precisely the mission of this deep dive. We are looking at some really recent research from 2023 where scientists studied the Anna's hummingbird, that's Calypt Anna, to figure out their specific behavioral and physiological responses when they drink nectar that has, low concentrations of ethanol in it.
And we should establish right away that this isn't some strange niche thing. This kind of exposure is completely unavoidable for them in the wild, isn't it?
Exactly. It's just basic environmental biology. Nectar is sugar water, and, fermentative yeasts are absolutely everywhere in nature.
Right.
They metabolize the simple sugars, and that yields carbon dioxide and, crucially for our discussion, ethanol as a byproduct. So for an obligate nectarivore like a hummingbird, a creature that has to drink these massive volumes of fluid every single day just to keep its metabolic fire going, this exposure to low-level alcohol is chronic. It's just part of their daily life.
And we already have some context for this from the mammal world, right?
Sure.
I know previous studies on animals like eyes and slow lorises show they often have a genuine preference for low-concentration ethanol.
They do. Sometimes up to 4 or 5%.
And the theory there is that it helps them find ripe, high-energy fruit.
Correct. But the avian response, that's largely been a blank page. The prevailing hypothesis, and the one they really tested in this study, was that, given how common fermentation is, these birds must have evolved a strong tolerance for low-concentration ethanol.
They just have to deal with it, so they must be equipped to handle it.
Exactly.
So if the hypothesis was tolerance, how do you even set up a test sophisticated enough to catch a tiny bird's subtle preference or, aversion? Let's dive into the specifics of the choice setup.
Okay, so the subjects were a handful of adult male Anna's hummingbirds, and they were housed in captivity. They were kept active and healthy on a standard 20% sucrose synthetic nectar.
And then came the experimental solutions, the ones designed to mimic what they might find outside.
Precisely. They took that 20% sucrose solution and they spiked it with 3 different concentrations of ethanol. 0%, which was the control.
Of course.
1% and 2% volume per volume.
And just to give that some context, how does that compare to what's out there in nature?
It's a great question. Some tropical palm nectars, which mammals consume, can naturally reach almost 4% ethanol, so 1 and 2%. That's a very plausible natural range, maybe even approaching a potential upper limit.
That binary choice setup is really neat because it forces a decision based purely on taste and preference. What was the critical methodology here?
It was really elegant. They used a 3-hour binary choice test. The bird was placed in a cage, equidistant from 2 small suspended syringes.
Okay.
And each syringe held one of the experimental nectars. So, for example, 0% versus 1%. They chose a 3-hour window because it's long enough for the bird to experience a full feeding cycle.
And the key metric wasn't just if the bird drank, but how much, right?
Absolutely. The metric was the differential mass lost from each syringe over that 3-hour period. If the bird preferred one, that syringe would lose significantly more mass.
Simple enough.
And to control for any kind of location bias, they even switched the placement of the syringes halfway through each trial.
Smart. Okay, let's shift gears to the data. After all that meticulous setup, what happened? What happened when the hummingbirds actually made their choices? Did they see that 1% ethanol as a nice little bonus, or did they reject it?
This is where we get the first really clear insight into their tolerance. When the birds were offered the choice between the unspiked 0% solution and the 1% ethanol solution, they showed complete indifference.
No preference at all.
None. They did not discriminate. And this finding, it strongly suggests that 1% ethanol is completely tolerated. It indicates it falls within a natural, acceptable background level in their diet that they don't even need to really factor in.
Okay, so that's the tolerance finding. But what about aversion? Did they find a concentration where the birds finally started to say no?
They did, yes. There was a very clear aversion point at 2%. When the hummingbirds were presented with the 2% ethanol solution, they showed a significantly reduced rate of consumption.
And this would have been compared to the 0% or the 1% options.
Exactly. And this aversion seems to be primarily mediated by taste.
Yeah.
Which, it makes sense. At that higher level, the alcohol flavor might just be too bitter or unpleasant to warrant the caloric reward.
To really break that down, we have to look at the numbers, because this shows us the actual physiological load these tiny birds are handling. Let's talk about their ethanol intake.
Yeah, the consumption data is absolutely critical for understanding their dosage. So when the birds were offered choices where they could opt for the less alcoholic solution, say 0 versus 1% or 0 versus 2%, their total ethanol intake was substantial, but it was surprisingly consistent.
How consistent?
It hovered right around 3.6 milligrams per hour in both scenarios.
But what about the extreme case? What if they're forced to choose between two solutions that both contain alcohol, like 1% versus 2%, where they can't really avoid it?
That's when the intake spiked dramatically. When the birds were forced into that position, the total ethanol intake rate shot up to a really striking 9.5 milligrams per hour.
Wow. So let's analyze those numbers, especially that 3.6 milligrams per hour figure. Does that number maybe suggest some kind of physiological limit they're trying not to cross?
The researchers propose exactly that. That 3.6 milligrams per hour rate likely represents a limiting threshold for these birds. The 2% solution seems to exceed their physiological comfort level, or maybe their toxicological threshold, which triggers that taste aversion.
Well, the 1% solution just sits comfortably within that metabolically acceptable range.
Precisely.
And this capability to process ethanol, this pretty impressive tolerance, is this a recent adaptation, or is this baked into their deep evolutionary history?
Oh, this is deeply evolutionary. It's intrinsically tied to their metabolism. The key to handling alcohol lies with two metabolic enzymes: alcohol dehydrogenase, or ADH, and acetaldehyde dehydrogenase, which is ALDH.
Let's spend a moment on those, because they really do sound like the heroes of this story. What exactly do ADH and ALDH do?
You can think of it as a two-stage detoxification process. When the hummingbird drinks ethanol, the ADH enzyme steps in first. Its job is to break down the ethanol into a compound called acetaldehyde.
Okay, and acetaldehyde is the bad guy here.
It's highly toxic. If it builds up, that's what causes the negative effects of alcohol, the flushing, the headache. So that's where the second hero, ALDH, comes in. It very quickly converts that toxic acetaldehyde into acetate.
And acetate is harmless?
It's essentially harmless, and in fact, it can even be used as a source of energy by the bird.
So the faster and more efficiently those two enzymes work, the higher the tolerance.
Precisely.
Yeah.
And what evolutionary studies show, especially in fruit and nectar-eating animals, is that these ADH and ALDH genes have experienced strong positive selection. Nature has favored species that can quickly detoxify that nasty acetaldehyde.
So the fact that the Anna's hummingbird tolerates 1% so easily is really powerful evidence of a long history of selection pressure to process fermented food efficiently.
It really is.
Okay, we know they tolerate it in the lab, but what about the real world? Did the researchers check the local pubs or, I guess, the local backyard feeders?
They did. This is section 3 of the study, and it's a great transition to real-world, human-influenced environments. They measured wild Anna's hummingbird feeders filled with that standard 20% sucrose solution located right on the UC Berkeley campus.
So what were the typical concentrations they found in that kind of backyard environment?
The ethanol concentrations in those feeders, which were, left out for typical amounts of time, they peaked at approximately 0.045% ethanol.
Oh, that's quite low.
It is very low, significantly lower than the 1% they tolerated in the lab, but, and this is the crucial part, it is consistently detectable. It's a non-trivial exposure.
And what drives that fermentation? We know yeast is present, but did they identify a strong link to external conditions?
The temperature connection was the big revelation here. The researchers found a strong and positive correlation between the average weekly change in ethanol concentration and the average weekly air temperature. And the statistical confidence was high. They got an R-squared value of 0.85.
And for you, the listener, R-squared means this. 85% the change in that ethanol concentration in the feeder can be tracked right back to the thermometer. Warmer weather means more yeast activity, which means more ethanol.
It's a beautifully simple, elegant ecological mechanism. The warmer it gets, the higher the ethanol level.
Now, this is the aha moment. because now we can contextualize the dosage against the hummingbird's sheer need for fuel. Their metabolic rate is legendary. A hummingbird has to consume about 10 grams of 20% sucrose solution daily just to meet its astronomical energy needs.
Exactly. And let's translate that chronic exposure into body mass specific terms. Even at a typical very low feeder concentration of, say, 0.02% ethanol, a hummingbird's daily dosage works out to 2 milligrams of ethanol. That corresponds to about 0.04% of its tiny body mass.
Right, let me process that. This tiny creature is constantly putting a relative chemical load into its system. Now let's compare that to us. If a human, say an 80-kilogram person, has one standard drink a day, that's about 12 grams of ethanol, what's our body mass exposure rate?
That corresponds to an exposure of about 0.015% of body mass.
That is staggering. So even at the extremely low concentrations in a typical backyard feeder, the hummingbird's body mass-specific exposure rate to ethanol is more than double that of a daily human drinker.
It is. This calculation, it just underscores why they need metabolic superpowers. Because they're so small and they drink so much volume relative to their size, a small dose of ethanol hits them proportionally harder and faster than it hits us. Without a robust metabolic capacity, those highly efficient ADH and ALDH enzymes, they would be perpetually impaired. Or worse.
So that 1% indifference we saw earlier, it really just confirms this metabolic requirement. They've been dealing with this fermentation phenomenon for eons, long before we ever put up those bright red feeders.
And this connects us directly to a really cool concept called hormesis. Hormesis is the idea that chronic consumption of low levels of a substance that might be toxic at high levels, can actually offer subtle physiological benefits.
That sounds completely counterintuitive. What kind of benefits are we talking about?
Well, researchers have explored this across species. For example, fruit flies exposed to intermediate low levels of gaseous ethanol have shown increased longevity and increased fecundity.
Meaning they reproduce more successfully.
Exactly. So this low-level chronic exposure to a mild toxin may stimulate detoxification pathways, potentially increasing overall metabolic resilience and even lifespan. The hummingbird's consistent low-level exposure might be doing something similar, just keeping those ADH and ALDH systems tuned up.
So fermentation isn't just a tolerated byproduct. It could actually be a subtle, long-term health boost for the hummingbird.
Potentially, yes. And ecologically, we have to grasp just how ubiquitous this is. Floral nectar starts fermenting the second it's exposed to microbes. Yeasts are widespread, and some research suggests that bird-pollinated flowers, which are often open and have large volumes of high-sugar nectar, they might have a higher incidence of these microbes compared to, say, insect-pollinated flowers. Unfermented nectar might actually be pretty rare in nature.
But the study did have its limitations. They were dealing with simplified, artificial nectars in the lab.
That's the main caveat. The 20% sucrose solution with pure ethanol doesn't perfectly replicate the complex chemistry or the mineral content, or, critically, the complex microbial communities you'd find in natural nectar or even in a real-world feeder.
So what are the next steps for research?
Future research really needs to zoom in on the specific structure and efficiency of the ADH and ALDH enzymes in birds, and more importantly, measure ethanol concentrations within truly wild floral nectar to confirm how this all plays out in the ecosystem.
Okay, so let's synthesize the core takeaway for you, the listener. What did these results tell us about the hummingbird's relationship with alcohol?
They drew a very clear behavioral line. Anna's hummingbirds actively reject high levels, specifically that 2% ethanol, which they sense through taste, but they readily tolerate low levels, up to 1%. And that reflects an evolved metabolic superpower that's required by their enormous relative intake. Their small size necessitates this robust tolerance, allowing them to metabolize alcohol at a rate that far exceeds a human drinker, even from a simple backyard feeder.
And this raises an important question that goes way beyond the feeder. Given how common fermentation is, does the hummingbird's evolved tolerance for ethanol actually influence plant-pollinator interactions? Might a slightly fermented flower be more attractive because the smell of that low-level ethanol signals a high-quality, high-sugar reward, just like it does for some fruit-eating mammals?
And if we look further down the road, how does this chronic low-level exposure affect their long-term health and behavior in the wild? Does that constant stimulation from hormesis provide a general fitness benefit, or are there subtle long-term costs to continuously running their detoxification systems that we just haven't identified yet?
Fascinating stuff. If you found this deep dive interesting, we definitely encourage you to explore the concepts of hormesis and avian ethanol metabolism further. Thank you for joining us for the deep dive.